Ladies and gentlemen, welcome to the Immunology lecture. Our agenda for today is to go over the different lines of defense of our immune system. A lot like a war, where a battle is continually fought between the environment and our own body's natural defenses. If you take your finger and your pointer finger and you look at this fingernail, it's about 1 square centimeter. If you touch around, table, clothes, you have don't touch your face. You have a total of 10 times 10 to the 1 times 10 to the 8th. In other words, a 1 with 10 with 8 zeros. I think that's 100 million bacteria per square centimeter, continually lurking around, ready to attack our body. And so we begin chapter 15. If you turn to page 568, the case of the recurring chemistry nightmare, it's a story of a person who has stress and develops herpes. In other words, herpes simplex 1, the fever blister from the stress that they have. And it kind of talks about how society discriminates due to illness. And I just had a friend who tested positive for COVID, and she's like, don't tell anyone because I don't want all the ruckus. You know, here you are with disease, and society often discriminates against people with disease. So that's the broader picture, psychologically, socially, of disease in our society. Our agenda, of course, today is not to really discuss that, but to keep that in the back of our minds about disease, but to discuss the immunology or the lines of defense against these invaders, 100 million per square centimeter around us. That doesn't mean that they're all bad. Many are normal skin flora, staphylococcus, that don't bother us. But to make a long story short, let's turn in chapter 15 to page 579 and look about the nonspecific immunity. And the first chapter parts of the chapter look at the skin as a natural line of defense. All right, the border skin that we talked about in A&P1. And as we move on, and you start with page 579, we'll talk about the first line of defense. The skin and mucous membranes are delicate in our nose, by our mouth, by our eyes, any of the open areas, and invaders mostly enter in those areas. So any of the holes in our body, that's why we have hairs by our eyes, by our mouth, by our nose, to try to prevent the invasion of enemies. So the skin is a tough physical barrier, though. You feel your skin, it's made up of mostly what? Keratin proteins. And the keratin proteins are a tough layer impregnating the epithelial cells and preventing really any invasion unless the skin is breached. It's a physical barrier. You can call it barbed wire in a war. Prevents things from getting in. The pH of the skin generally is a bit acidic. 3 to 4 is its acidity, and that drives away microbes. Most microbes don't like that acidity. Saliva, tears, and mucus are all still part of the first line of defense, where we have little antibodies. We have something known as lysozymes in the saliva, tears, and the mucus that wash away the invaders. These are antimicrobial agents. So when you have tears, they wash away the invader. It's not a bad thing. And let's take our large intestines. We have a lot of bacteria, in other words, a whole flora or community of bacteria in our large intestines that defend the home territory from water coming in and things coming in that may be infected. We have our own set of bacteria that defend and actually give us vitamins, and we'll talk about that more in the digestive unit. There are guard dogs or our pets. So the intestinal bacteria have their own protective element. Stomach, very acidic. You want an acidic stomach to destroy invaders that come in. The food we eat is filled with bacteria. You know, it's just objects around. You eat a piece of bread, there are bacteria and things that are on there. The stomach acid destroys, breaks apart cell junctions of these invaders. Very important in protecting our body. Cilia, we talked about hairs. Cilia are little hairs. They beat in the respiratory tract in outward and push out microbes to protect our lungs from getting invading creatures, viruses, bacteria, etc. And that's all part of the first line of defense. That's where you're not involving your immune system. It's protective. You know, they always say an ounce of prevention is a pound of cure. All these things are protective. You don't even get into the body, with the pH of the skin being low, with the intestines having their bacteria, the stomach having its acidity, with the saliva and the tears, with the mucus and the hairs of your respiratory tract. It's supposed to keep out a lot of these invaders, because you don't want to resort to the second line of defense, which is the inflammatory response. You know what an inflammation is: swelling, heat, pain. When a microbe invades or somehow physically gets into the body, cells are injured. We say there's cell necrosis or cell damage. We talked about this immune response in ANP1, but what I'd like to do is go over the main chemicals of the immune response. One, histamines. When you're in the second line of defense, the inflammatory response, histamines are given to vasodilate the capillaries, which in the last lecture we said brings more blood flow to an area. Prostaglandins also do this, vasodilate. More blood flow to an area means more white blood cells going to an area, which means more protecting cells to an area. Heparin is also given off. That thins the blood. Why? Why would you want to thin the blood in an area that has been damaged? Because your troops, or your what we call white blood cells, those are your tanks, your protective troops, have to be able to move to attack the enemy. And the thinning of the heparin allows that. This whole idea of increasing transportation to an area makes common sense, right? If Canada invades Maine, which seems ridiculous, but you're going to send troops not to Florida, right? You're going to send troops to Maine. So in an area that has been breached by some sort of invading microbe, you've got to get more troops to that area. So you vasodilate the capillaries using histamine and prostaglandins so that more troops or more protective cells get to the area. We say granulocytes come to the area. As you know, those are the neutrophils primarily. Basophils and eosinophils also come to the area first. So neutrophils, let's go over, are white blood cells that are the first to arrive in an area during a bacterial infection. If you look at figure 15.9, it shows a breached area with invading organisms and granulocytes coming to that area. If you look at table 15.1 on page 581, again, table 15.1, it shows a picture of the neutrophils, the basophils, and the eosinophils, and the monocytes. So take a look at those. Those are your tanks. That's the third line of defense, the tank line of defense. I call it the chemical warfare. Involves your tanks and involves chemicals. So let's talk about these tanks. The tanks are your white blood cells. Neutrophils come to the area first and are bacteria slayers, using that respiratory burst that we talk about to attack the enemy. Basophils are more found in causing allergies. Eosinophils more or less go after parasitic infections like ringworm, malaria. Monocytes, on the other hand, are a very large white blood cell. If you take a look at a monocyte, it will slowly get developed, takes a little bit of time, unlike the neutrophil, to get to the area, and it becomes larger. And the larger macrophage is pictured in table 15.1, and it gobbles up or phagocytizes the enemy. It's slow, it's large, and it gobbles up and forms like giant cells. That's an agranulocyte. That means it does not have little granules in its cytoplasm. These are your big tanks that destroy the enemy, right? Notice I have not mentioned the lymphocyte. That's going to come later. from table 15.1. If you look on figure 15.10, okay, phagocytosis is depicted, where you can see how antibodies and little bacteria are getting gobbled up by the enemy. That's phagocytosis. Again, figure 15.10. So you can see it happening in action. Interferons, your third line of defense, involves the chemicals. The chemical warfare, whoops, why that happened. The chemical warfare of the third line of defense involves pyrogenes, complement, and interferon. Let's go over interferon. When a cell is dying, it will, because let's say a virus has invaded it, it will give off antimicrobial enzymes known as interferon that prevent the next virus from getting into its nucleus. It prevents what we say the virus's RNA from becoming protein and taking action against the enemy. Interferon stops viruses. It's a natural cell defense against viruses. And interferon activates phagocytes as well. We say that it brings interferons attract more white blood cells to the area. In other words, activates phagocytitis. This is called cell altruism, because altruism means giving of yourself at the expense of yourself to save someone else. So the cells that are dying give off interferon to protect the cells around it. So they die, but they protect the cells around it. So that's the way interferon works. Pyrogenes work by increasing temperature. You increase temperature, you actually decrease microbe growth, because microbes want to grow at our body temperature. So if you bring it up to like 99, 100, you prevent the microbe from growing. Then we have complement. Complement is not, those are nice shoes. Complement is a series of about 20 different proteins that act together in a cascade to lyse or blow holes in invading microbes. They destroy the invader. A complement cascade, we call it. So on these pages, 581, 582, 583, I give that line of defense, the white blood cells and the chemicals that are associated, pyrogenes, interferon, all of that is discussed, up to 584. On page 584, we start this last line of defense known as the specific immune response. The fourth line of defense, specific immunity, can be compared to what we've talked about all along. If you look on figure 15.2, everybody, the skin is your first line of defense, right? The inflammation, your second line of defense. Fever, interferon, phagocytosis. All of this is very general, non-specific. It's written as non-specific immunity. Only in this last line of defense, known as humoral and cell-mediated immunity, is it a specific line of defense in figure 15.2. And that keeps you alive. So let's look on page 584, all right, page 584, the difference between cell-mediated and humoral immunity in this specific line of defense. Humoral immunity involves the humors of the body. The word humors mean liquids of the body. And B cells, this should be cells, that should be an E. B cells of the body produce antibodies. Antibodies, as you know, in the last ANP1, are little airplane-shaped proteins that attack antigens, which is a fancy word for foreign substances. And the humoral, the humoral immune response involves antibodies produced from B cells that attach and destroy antigens. These are the 5 types of antibodies that are produced in our body. And I want to say that you should be able to match the different types with each. Like IG is another word for antibody. So IgE is involved in allergies. IgA is found in tears, saliva, and milk. So let's stop there. If anyone have an allergy, raise your hand. That means your IgEs are getting activated. when babies nurse, they get milk from their mama. So immunoglobulin A's are directly given from mama to babe, and that is a big benefit of nursing. Immunoglobulin M is the first made and the last, they only last a short time. Immunoglobulin G's are involved in activating the complement that we just talked about, which blows chunks in the enemy. They agglutinate or clump antigens together. We don't know about IgD, but we know that they generally attack antigens. This is the structure of the antibody. It has a dark chain in the center. We call it a heavy chain. That's this and this. That's the center of the plane. And the outer parts of the wings are the light chain, okay? The tips of the wings have what we call a variable group. That means a group that is different for each type of antigen. So the V variable group for COVID-19 is very different for the V variable group for chickenpox. All are the same in terms of their C areas, their constant areas. Last but not least, disulfide bonds, these little S's, hold the antibody together. So the antigen binding site is always the wings of the antibody, okay? And the antibody works, if you remember, by precipitation, agglutination, neutralization. If you look at figure 15.16 and complement, those four methods you've got to know, where neutralization neutralizes the enemy, and on figure 15.16, again, agglutination clumps, precipitation brings it out of solution, and complement blows holes in the enemy. Know that chart, figure 15.16, because those are the different methods of how an antibody works. Last but not least, in specific immunity, is how the T cells work. What's a T cell? Well, you have B cells and T cells. B cells make antibodies. T cells are involved in direct hand-to-hand combat with the enemy. They are special forces ops. They are not your tanks. Your tanks are your monocytes and your neutrophils. But these are special tanks, special white blood cells that derive, both B cells and T cells derive from lymphocytes, and they are the brains behind the operation. They are depicted in figure 15.15, okay, the different types of T cells, and they are shown, of course, in our table 15.1 on page 581. Those are T cells and B cells, and the thing to note about them is that the B cell makes antibodies and circulates around as little plasma cells, also known as B cells, circulate around and make antibodies. And when you have long-term immunity, like let's say you get immune from the coronavirus, that means you have what we call memory cells. Plasma cells that stick around a long time and continue to make antibodies to prevent future infection. That is memory cells. Cell-mediated immunity is mainly based on T cells. T cells, like I said, are special ops forces. They are special white blood cells made from the lymphocytes that directly attack the enemy. And if you turn to figure 15.17, how does cell-mediated immunity work? Well, it starts with the macrophage back on the second, third line of defense, where the macrophage gobbles up the enemy. And what it says is, here, T cells respond to receptors or proteins presented by the infected host cells. So if a cell is infected, the cell will present some of the enemy structures on its surface. But mostly, if you look in figure 15.13, the macrophage gobbles up the enemy and takes a piece of the enemy and puts it on its cell surface and says, here it is, I'm macrophage presenting. In other words, here it is, this is the structure of the enemy that you need to look for. So in other words, if it's the coronavirus, then here we are, the macrophage puts a piece of the enemy on its cell surface and tells all the rest of the immune system that that's who it should go after. And if you look in 15.17 in that figure, the macrophage presents the antigen, The antigen is the little triangular shaped structures, and that antigen attaches specifically to a T helper cell. And the T helper cell, all right, initiates what we call the rest of the specific immune response. Why don't we go over that right now? If you turn to page 590, you'll see that the T helper cell gives off lymphokines to stimulate antibody or humoral immunity and cell-mediated immunity. Antibody immunity is very simple. It just means that B cells are going to be turned into plasma cells, and plasma cells, as you can see on figure 15.17, make antibodies that attack the enemy. If you look on the left side of the chart, it's cell-mediated immunity, where it involves the activation of T killer cells. How do you do it? Well, the macrophage presents, all right, and then B cells are activated to make antibodies, but cytokines are released to cause T helper cells to bind to the macrophage that's presenting and release interleukin 1. Interleukin 1 is a chemical that causes more T helpers to come to make interleukin 2. Interleukin 2 causes T helpers to divide more quickly. So what am I really saying with all this? These chemicals, interleukins, get the T helpers to divide, and then the T helpers make cytokines that cause, here's the key, the special ops guy to come in, cytotoxic T cells, which blow holes using perforin in the enemy. My favorite picture of all is a picture of the enemy being destroyed by the cancer cell being destroyed by a T cell. And if you look on figure 15.14, that's a little cartoon of it where or actually a micrograph of the T killer cell, in other words, the cytotoxic T cell killing the cancer cell. Direct evidence of the enemy getting destroyed. And you look at figure 15.13, that is a picture of the macrophage presenting, okay? How it gobbles up the enemy, presents it on the cell surface. Figure 15.15 shows another picture of how humoral immunity happens, how the T helper cell stimulates memory cells and B cells to produce antibodies. It also shows how interleukin causes killer T cells and other white blood cells to get activated and continue everything going. And everything goes wild, attacking the enemy, until the last cell comes in, T suppressor cells that demilitarize and turn off the immune system after an infection is over. And this is my cartoon of the entire process, right? Let me go over this again because it's hideous to visualize, right? You've got the book to help you visualize. You have this lecture and the notes, and now you've got the cartoon. Let me go over this cartoon again. It's going to be repeating just what I said. You don't have to listen if you understood it really clearly. Take a look at this. The macrophage presents when it comes into contact with the enemy and phagocytizes it. So let's say it finds the coronavirus, gobbles it up, presents to the enemy, and T helper cells come in that attach specifically their receptors with that viral protein or whatever protein it is from an enemy. That causes B cells to attach to specifically T helpers. And it causes B cells to divide, become plasma cells, make antibodies, which attack the antigen, and make memory cells that, as soon as an enemy comes in, will remember, that's why it's called a memory cell, it will remember that enemy, form plasma cells and antibodies to attack the enemy. That is all known as humoral immunity. See that? That's humoral. The humors of the body defend you. Cell-mediated immunity means hand-to-hand combat. Cell-mediated means hand-to-hand combat. So cytokines are released to attract cytotoxic T cells, and the cytotoxic T cells gobble up in hand-to-hand combat, boom, 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 and blow holes in the enemy. The T helpers also give off interleukins that attract more T helpers. That's, just positive feedback. All of this gets turned off by T suppressors to turn off your whole immune system. So that is the cell cartoon of specific immunity against an enemy. Hopefully you've enjoyed that. Last but not least, we're going to talk about something very important in today's world, types of acquired immunity. Natural versus artificial. Okay, how do we get immune from things? I mean, that's the big question with the vaccines right now that we talked about with the coronavirus versus getting natural immunity. So active acquired immunity versus passive acquired immunity. So two types of immunity. How do you get immune to things? Active acquired immunity, the word active means your own immune system activates itself to take action and make memory cells against enemies. And the natural active acquired immunity is just you get sick and then you're immune. That's what we call, what you always hear of, herd immunity. If enough people, over 60%, 60 to 70% of a population just gets natural acquired immunity, population is said to have herd immunity, and the virus is unlikely to pass around all that much in that society. Obviously, the more people that get sick, I hate to say it, the less likely it is that you're going to get sick. So go out and get sick. It all depends on how you look at it. If people get sick and become immune to it, we say that the body's own immune system has produced its own memory cells to immunize the person after an infection, like the chicken pox. You get the chicken pox, now you're immune. That's your natural active acquired immunity. Artificial or synthetic acquired immunity that's active is a vaccine. You get a coronavirus shot when it comes out, and that's a dead piece of the antigen, either a dead piece, a weakened piece, or just a piece of the antigen is put into the body in a vaccine. Now, they're not using weakened, they call it live attenuated. We're not using weakened forms of viruses anymore. Generally, we use a piece of the virus that's dangerous, the spike protein in the case of the coronavirus that causes COVID-19. And we have, therefore, no danger of actually getting the disease, but we activate memory cells to and plasma cells to produce antibodies against that virus. And as of this moment, which is July 15th, 2020, Moderna has just had a vaccine that has stimulated an excellent immune response, a good amount of a good amount of antibodies was stimulated by that new vaccine. So it looks promising as of this moment. I can't predict the future. So that's artificial active acquired immunity. Now, what's passive acquired immunity? Passive means you're not doing much. Your immune system's not getting activated to take action to get you immune. So natural passive acquired immunity is breastfeeding. Definitely antibody A and immunoglobulin A is transferred from mother to infant in milk. That is natural passive acquired immunity. Artificial passive acquired immunity is when you just get plain injected with the antibodies. That's another way they're searching to get a vaccine, by just shooting you up with antibodies that are made by gene technology, just producing the antibodies and giving it to you. Now, rabies works that way. The rabies vaccine doesn't get you activated actively. It just passively gives you antibodies to fight the rabies or rhabdovirus that has invaded you through a bite. So interesting. That's artificial because you're getting shot up with something, but it's passive because you're not really making the antibodies yourself. You're being given it. So that's the types of immunity, the 4 types. Be sure to be able to identify those 4. Fascinating, right, for our times. Last but not least, how does your body know that a virus or bacteria is not itself, that it's different, that it's an enemy? And that the way it recognizes self from non-self is by something we call the major histocompatibility complex, or M8. That's your biggest word in anatomy. Histocompatibility. Histo means tissue, compatibility. How compatible is the tissue? MHC, we call it. These are sets of glycoproteins on plasma membranes that code for, that are special for each of us uniquely, all right? MHC is different for each person. It's coded for over for by about 20 different genes. And when you do tissue transplants, your MHCs have to match the donated tissues in order for your body to not reject itself. So my point is, your body at an early age recognizes self from non-self by detecting and identifying different MHCs. If the MHCs are different, so a virus comes in, it has a different protein, or you get a liver transplant and it's a different MHC, there's tissue rejection. Your own immune cells will recognize it as non-self. And recognizing self and non-self has a lot to do with allergies. If you're a kid, you've never been exposed to pine needles, your body at an early age recognizes self and non-self and what's good and what's not. And the immune system tends to form allergies when it has not been exposed to that type of MHC at an early age. Some even argue that clean kids have more allergies growing up clean when they're older, more allergies because they haven't been exposed at an early age to determine self and non-self. The scope of how allergies develop is beyond this course, but I just wanted to give you a little taste of it. Something weird, mice outbreed with respect to MHCs. What does that mean? A mouse goes up to another mouse, wants to mate, it smells each other, they don't even know if they're too closely related, and then the mouse runs off and says, whoa, I smell you. They can smell the MHCs and they determine, I don't want to go near you, you're probably my brother or sister. So it lessens inbreeding. Isn't that a weird thought? It lessens inbreeding. So mice outbreed with respect to MHC because inbreeding causes a lot of genetic problems. Class 1 and Class 2 MHCs. All cells of your body have Class 1 MHCs. Liver, kidney, brain, all Class 1. Class 2 are only on your macrophages and B cells. Those are your immune type cells that have Class 2 MHCs. So just wanted to differentiate them. Hopefully you've enjoyed this lecture on immunology, and our exam will be in the next class. Okay, thank you and have a pleasant day. And for the summer A&P class, that will be next next Wednesday. Okay.